The gravity of the time is such that every new avenue of peace, no matter how dimly discernible, should be explored. Never before in history has so much hope for so many people been gathered together in a single organization. You will provide a great share of the wisdom, of the courage, and the faith which can bring to this world lasting peace for all nations and happiness and well-being for all men.
So we are here today with Dr. Lindsay Morton, who's the Deputy Chief Science Investigator at the National Cancer Institute, which is part of the National Institute of Health. Lindsay, welcome to Titans Nuclear.
Thanks for having me.
Yeah, so we'd also love to learn about your background, where you grew up and how you got into the field of medicine.
So I grew up in the Midwest. I'm a Midwesterner at heart with family from Illinois and Michigan and Wisconsin, spent a little time in Ohio, but I went to school out on the East Coast, and I feel incredibly fortunate. So I'm trained as an epidemiologist, and that's a field that I certainly had never heard of growing up.
And what does epidemiology mean?
Exactly. So now, actually, these days, because of COVID-19, unfortunately, many more people know about epidemiology than ever did previously. So we think of epidemiology as the study of population level health. Mm-hmm. I often think of it as the who, where, what, when, and why of disease. And we take information and kind of aggregate it across among many individuals and take that collective information in order to try and better understand who gets diseases, what diseases do they get, when do they get them, where, because there are different diseases that are more common in different populations than others. And then, of course, why they get different diseases.
And is this mostly data driven? Is it, and let me ask that even more specifically. Can you pull insights out of just the raw data alone, or do you have to also have some thesis going in to kind of test against the data?
You can do both, actually. It depends a little bit on what research question you're asking and how you want to approach the data. And then you have to design your study properly, depending upon whether you're going in with what we call the agnostic look, as opposed to testing looking for a specific relationship.
And what interested you to epidemiology specifically?
Yeah, so that's what I was going to say. I was so fortunate to find out about epidemiology. I was a sophomore in college and working as a research assistant for this wonderful biology professor, Tom Roos, who since passed away. I was at Dartmouth College and just getting a great liberal arts education. And I was talking with him one day and just said, I don't know what to do with my life. I don't know what career I want to pursue. I really love medicine, but I don't know if being a practicing physician will make me happy. If that's really what I want to pursue. I love biology, I love math, and I love research. And I'm not sure what I want to do with my life. And he said, well, have you ever heard of epidemiology? Because you essentially just defined it. And it really was that love of math that integrated into my interests that led me to epidemiology. So I started out, this terrific professor said, look, I've got a friend who works actually at this coalition of free health clinics in Vermont, just across the river. And essentially they had a database challenge at the time, and they were trying to make sure that they were doing as good a job as they could at serving the patients that came to their clinics. But they had a challenge about linking information from the different data sources. And so this is kind of a classic data management, numbers, and health question. And so I started to get into it, and it just all really resonated with me. Totally different part of my background. I was a swimmer. I've always loved the ocean. Like a lot of kids, I wanted to be a marine biologist when I grew up, when I was much younger. And I had taken an off term at Dartmouth and actually done a little bit of work with Earthwatch, volunteering with dusky dolphins on the east coast of the South Island of New Zealand.
Wow.
And so I had the opportunity to design a project for my senior year in college. It was called a senior fellowship. And I designed an entire research project related to epidemiology. And I designed an entire research project related to dolphins. And was talking with the same professor who was a great mentor and said, I want to, we had agreed I would apply for this fellowship and he would be my mentor. And we were talking about the pros and cons of the different projects. And he said to me, Lindsay, what are you going to do? And I said, I'm going to become a cancer epidemiologist.
Okay.
So I should do the epidemiology project, shouldn't I? And he said, no. He said, you should go study dolphins in New Zealand for a year and do everything that you need to do in order to learn about the research process. You have to develop the idea and the hypothesis. You have to design the study, collect the data, analyze it, write it up, and present it. And he said, if that's what you learn from the senior fellowship, you'll be well on your way in your career. And then you'll have an entire lifetime to try and improve people's health.
Wow.
So I did. I went back and
Good advice, yeah.
Out in the field and out on the water, collecting data on dolphin pod movement and mating behavior in Kaikoura, and then went on to my career in public health.
Is there anything that you draw from that experience, not just having gone and done that, but literally from dolphin behavior and watching and studying them that you've applied to any other field of science you've looked into? Is any coincidences there?
I haven't. I think that it really cemented the idea that I want to improve people's health. I'm very passionate about conservation issues and think that the work in that area is really important, but I wanted to come back to people. And so it taught me a lot about myself, honestly. It also taught me a lot about my interests in the way that we measure different things. And as a scientist, we think about measurement all the time. We think about how we measure things, what exactly we're measuring, and the strengths and the weaknesses of our measurements. And of course, there different fields take different approaches to measuring things. But one of what I'm drawn to in epidemiology, and this is relevant to some of the work that we're going to talk about today, is being drawn to some of the precision of the radiation exposure metrics that we have. And I really appreciate the understanding the strengths and the weaknesses of the radiation exposure metrics that the physicists have contributed, for example, in our studies of Chernobyl. And that was something that I really understood early in my career, shifting from animal behavior to epidemiology, is that there are just different ways to measure things, and that matters a lot to me.
And why cancer as opposed to some other, so like we hear about epidemiology with virology right now. Yeah. So why cancer specifically?
Absolutely. So you apply the tools of epidemiology to any health outcome. And I chose cancer because, like so many people, I know individuals, loved ones who've been affected by cancer. Mm-hmm. And it just seemed like that was a real opportunity, again, to try and improve people's health on a very broad scale. I obviously really appreciate what physicians do. A patient walks in the door and the physician is responsible for really trying to address the health issues of that individual patient. And part of what epidemiology brings is the power of looking at larger numbers, and then you're a step removed, but potentially having a positive impact on a larger population. One is not better than the other. This is a theme that I would say is kind of throughout my entire career, is recognizing that different people are needed to be part of the scientific team to carry out research, and one position is not better than the other. They're all really needed in the scientific process, but you kind of have to figure out what suits your own strengths and interests.
And were there particular types of cancers that you focused on throughout your career?
Yeah, so I've used some kind of basic principles with the fellows that I work with. I'm always trying to share this advice that as scientists, we work hard, long hours. We're thinking about ideas all the time. And so you really have to be driven by passion for the topic that you're studying. So I started out actually studying blood cancers, hematologic malignancies like leukemia or lymphoma, for a very basic reason that I found the immune system to be fascinating. People are often looking for a much more sophisticated reason than that. And actually, it's a really important drive when you just find something to be really fascinating. We don't completely understand the immune system. There's so much left to be learned. And so when I started my doctoral program in cancer epidemiology, I expressed this interest in the immune system to some of the professors I was working with. And they said, why don't you start out by studying lymphoma? Sounds like a great plan.
And when you say immune system, is there like an organ that's responsible for the immune system, or is it really just cells in the blood are the immune system?
It's a little bit of a combination. So it's the cells in the blood, absolutely, but there are other lymphoid tissues. You can think of the lymph nodes or concentrations of lymphoid tissue, for example, in the GI system. And so it's complicated and has a lot of different types of cells, pretty much in almost every area of the body.
And when you say lymph nodes, like I think about, okay, like do I have swollen lymph nodes when I'm sick, and that's like in my neck. And but then you said GI, then I think of like in the intestine. Are there like little bubble pockety node things spread throughout the body?
You can find lymph nodes throughout most of the body.
Yes.
And what do they look like?
They really do. I mean, honestly, you can just look them up online and you can see beautiful images of chains of lymph nodes throughout the entire body. And they really do kind of look like little kidney beans, and they're different parts of the lymph node that have different jobs. The basic principle is that there are different immune cells that transit through the lymph node, and all the different cells, it's kind of like a gathering place, and the different cells have different jobs in the gathering place. So some cells are there because they're actually showing on their cell surface that they've encountered what they think is something foreign in the body, and they want to show that little foreign piece to other parts of the immune system and say, do you know what this is? And then if another cell recognizes that as a foreign, I'm trying to say the word peptide, right? But a foreign piece, essentially, then it can stimulate an entire immune response.
Wow. Okay. And now maybe we can kind of bring you guys together and learn a little bit about, where did your paths intersect? Is this the first project that you've worked on together, or have you been bouncing around the same building and bouncing ideas off each other for a while?
The latter. I mean, we've, I don't know, working together now 15 years, different kinds of projects and things. And, where we work is a very, very remarkable place that really focuses on what we call team science. So we bring people with different kinds of expertises together and work in teams. And most of the papers that are published by the division are team science, where you have multiple people working together. That's really essential. So these different expertises have different perspectives, and it helps to really enliven, but more importantly, give really important insights.
I love that idea of team science. And then, so how do the projects originate? Is it a grant process? Is there a big brainstorm session? Is there here's some top down from Congress, here's some areas that we're interested in, now go spend this money? Like, how does it happen?
Well, it's a terrific question. The National Institutes of Health is a very large funder, and it's made up of 27 institutes and programs. The National Cancer Institute is the largest of those, and within the National Cancer Institute, which has a very large budget, about 85% of the money is sent out in grants that people all over the US and some in other countries are able to compete and have funding to be able to do cutting edge science that's peer reviewed. A certain amount stays in the intramural program, and we're one of the 2 intramural programs where we are given a budget per year. And then we have 71 senior scientists and a number of other experts who are part of our group. And then we look programmatically and decide what's a high priority, such as studying the genomics of survivors from the Chernobyl accident. And the advantage of the intramural program is that we can think strategically over a long period of time. So the studies that you want to talk about with Chernobyl, we have investigators who, going back to the early 1990s, had identified the epidemiologists or went there and started to organize and follow individuals over an extended period of time, because we needed to know what the time course would be in which people would develop problems such as cancer or cataracts or heart disease. And we're particularly focused on cancer. So our funding is really focused on high priority issues. We like to think of ourselves as sort of doing the people's epidemiology. We do a lot of things that are difficult. We study a lot of subjects that aren't popular, but they're important to know about contaminations in the environment, whether they're radiation or from other kinds of things like pesticides or occupational exposures. I mean, those are the kinds of things that we need to know what those true cancer risks are. That's one of our major goals.
That's okay. That's a great segue into my next question. So radiation, what is the mechanism by which radiation causes a cancer?
Yeah, so part of what was so exciting about the thyroid cancer genomic sequencing project that we did was that this was one of the first opportunities to actually ask that question using the cutting edge genomic advances that have occurred in the last few years, as well as all of the incredibly rich data that were collected by the Chernobyl Tissue Bank and a number of collaborators. So, of course, we've known for years that ionizing radiation is a carcinogen. There have been tremendous number of animal studies and studies just in cells trying to understand what damage is caused by ionizing radiation and how that might cause cancer. But it's been really difficult to actually link that to specific human tumors. So I can give you
why. Why is it difficult? Like, why is one mammal so different than another mammal in the way that ionizing radiation might? I don't know. What does it do? Does it break apart a DNA strand?
It does. So it has a variety of different impacts, and this is known from a field that's called radiobiology. So it's been known for a long time that ionizing radiation causes a variety of different types of damage in the cell. It's long been thought that the most important was a breaking of both strands of the DNA. So, and that those strands either remaining broken or being formed back together, perhaps in error, could cause cancer. But there are other types of damage as well that are still being characterized. So some of that is more clustered damage or individual bases or just single strand damage. And because, again, I was mentioning the advances in genomic technologies, we're still learning how cancers actually develop, then it's difficult to link that actually back to the exposures. A lot of the research that's been done trying to understand the process of cancer development and all of the different genomic damage that's characteristic of cancer cells actually has not included any data on risk factors. So it's included clinical data or pathology data, but not the risk factor data. So that was one of the exciting things about the study that we did, where we were able to bring together the exposure data and all of the genomic data.
And still on that point, though, not to sound like an evil guy or something, but can't we just like put a bunch of mice in a cage, blast them with some radiation, and then look at them under a microscope to see what's happening? Is that crazy?
Stephen, do you want to talk a little bit about animal models versus humans?
Okay. You can do it, and there is a long history of doing that. But not all radiation is necessarily the same, so to speak. The reason I say that is how much and in what way you get it may be very important. So animal studies have taken rats or dogs and just buzzed them at very high doses. And we've learned some important things from that. But in the human situation, what was really remarkable is this is the first time where we knew from the Chernobyl accident from our health physicists and, our remarkable colleagues, in Ukraine and elsewhere, exactly how much exposure they had over what period of time. So we could actually map the genetic changes we saw back to discrete protracted period of time. It wasn't just standing in front of the machine and getting buzzed. We know from other studies where people have developed cancer after receiving radiation, therapy, that there are certain features, and they're similar to some of the ones that we've seen, but not always identical. So it tells us that it's not an either or, comic book like model that sometimes it gets portrayed as. So how and in what way people get the radiation may be particularly important. Okay.
And now our audience is very familiar with the types of radiation, alpha, beta, gamma. Is that what you're talking about in terms of the differences in different type of radiation?
Among other things, I think there are different types of radiation. And here we were particularly able to look at I-131, iodine, which in this setting, that was a substantial fraction of what was released in Chernobyl, and it fell in the environment, got into the grass, so the cows started to eat the contaminated I-131 grass, concentrated it, and then the milk served to the young kids. Because remember, this was the Soviet Union, 1986. You didn't have Uber Eats. They ate only, they had very restricted diets that were were sustenance diets, but not with any real variety. And so milk was a critical factor. And these children who we studied were drinking lots of milk around that time when it was contaminated. So, we can start to understand how the model of the exposure occurs, and then what that actually did in the genome. This is the first real look in humans for understanding what we know. Ionizing radiation causes cancer. It's an International Agency of Research on Cancer. IARC monographs have established this as a class 1 type of carcinogen. We all know it can cause it. There are things that radiation can do that are very therapeutically very helpful, but it can also be devastating. And that's that wide range. And now we're in this study beginning to understand what those effects could be that lead to specific cancers.
And as the result of the Chernobyl accident, other than the local radiation-induced harm, from the cleanup workers, is this the extent of the harm that was caused? Specifically the iodine 131 exposure to children? Or because we know that there are a lot of radioisotopes that were emitted. We know a couple of them can find their way into biology, such as strontium and cesium. Have we effectively narrowed it down to it was just the iodine 131 that ended up causing the cancers?
So I think it's a little bit more complicated than that. The reason we're so focused on I-131 in this particular study was because of the concentration in the thyroid. So when we're thinking about that particular exposure,
and that's my question. The very act of it spreading across, half of Ukraine and Belarus, in essence, dilutes things, which makes them less toxic. But the ability for it to reconcentrate through the grass mechanism, and then once again reconcentrate in the thyroid, is that the unique characteristic, or is that characteristic unique to iodine 131 that allowed it to become a potent carcinogen?
I think that is the description of what happened with the I-131. But when we look at our exposures, look at the exposures in the studies of long-term health effects in the cleanup workers, we don't just account for the I-131 exposure. We look at other radioisotopes and external exposures.
Yeah, the ones who are very local. I was thinking more about all the stuff that got spread out. Yeah.
More broadly in the residents, and we think about our studies of the adverse effects of the Chernobyl accident in these two categories of what happened with the cleanup workers versus what happened with the residents.
The rest of the public,
Fairly quickly, the focus was on thyroid cancer in the young children, but it's really important that we continue to monitor these populations and in fact have ongoing collaborations and studies where we're looking at other health outcomes in the residents as well as in the cleanup workers.
And how are we able to map the exact exposure of any given individual?
So right after the accident, the Ukrainian government actually and Belarusian and Russian governments actually made a number of measurements of radioactive iodine, so direct thyroid measurements in the residents and the surrounding areas. And so the team of radiation dosimetrists, so the physicists who are collaborators on the project, have worked together to extrapolate information from a combination of different sources, including these direct thyroid measurements.
And the kids that did end up getting cancer, what was the dose that was imparted on them that triggered that?
There was a whole range. So one of the important aspects of our analysis is that we were actually able to do what we call a dose response analysis. So because individuals had different doses and those doses had been reconstructed, we could actually model the dose level in relation to the level of the genomic characteristics.
So what was the range, the upper bound and the lower bound of people who got cancer?
Well, in the study, from individuals who were unexposed, because we included unexposed individuals who developed thyroid cancer in our analysis
as a control group,
comparison group, to individuals who just had, a few milligray, up to individuals who had more than 1 gray estimated radioactive iodine dose to the thyroid. But Brett, I was going to say before, as you were talking about different types of radiation, I also wanted to point out that we can talk about this more when we think about the implications of this study, but you were asking about the different types of radiation, and that this audience might be really interested in understanding that in our broader studies in the radiation epidemiology branch at the National Cancer Institute, we consider a number of other effects as well. So it is the type of radiation, and here we're focusing on radioactive iodine. I've already mentioned with the cleanup workers, we take into account their more complex radiation exposures, but we study patients who were exposed for both diagnostic and therapeutic medical irradiation. So we think about, it's not just photons, protons, we also think about dose rate, and the duration or the fractionation when we start to think of therapeutic exposures. What was the fractionation? What were the fractions of their exposure? So maybe a patient received 30 gray to a particular body region to treat a tumor. So it's kind of how much of the body is exposed and to what dose level, what type of radiation, and at what dose rate. And we try and account for all of those things in our radiation epidemiology studies.
And then
Can I add one?
please.
Brett, I think it's also important to recognize what we're studying here in Chernobyl are those people that have survived to a certain point. We know that when the accident occurred, there was very high exposure for some who died of acute radiation sickness. They developed leukemia or aplastic anemia, loss of blood cells in a relatively short period of time. In the chaos of the first few years afterwards, there was not a structured way to capture those. And that's very important to recognize. We're looking at individuals, some of whom had very high levels of exposure, but that they've survived and they've gone on. And the question that they then subsequently develop the most common type of cancer in younger individuals who see I-131 thyroid cancer, the papillary thyroid cancer. But we're not able to capture all of the cancers that occurred due to this. And this is an ongoing controversy that I don't think anyone will ever be able to fully answer. But we fully recognize that there were many things that occurred around and shortly after the accident that clearly led to cancers that we can't quantify simply because we don't have the information the way we do with the survivors. Okay.
You're saying among the cleanup workers, those are the ones that are harder to quantify.
Or even the evacuees, the people that lived there and remember, it took a period of time before the Soviet government actually recognized and mobilized. So you had individuals who had days of quite extensive exposure in Pripyat, in that nearby region. So
exposure to which radioisotopes?
Well,
still iodine-131 or others?
Others as well. And so again, that's a group that we have not been able to study as well. So I think we have to recognize that we've been able to set in at a certain point, but there was a degree of exposure. And this is very important in both the Ukraine and the Belarus as they continue to think about this and how and in what ways they both honor and recognize those things, those health consequences that did occur directly due to the Chernobyl accident.
And specifically with thyroid cancer, one thing that I've read is that sometimes like they'll detect nodules and then they won't become malignant. And so now there's like a practice amongst doctors to actually not like go in and do biopsies, even if they feel that the nodule. How do we tell the difference between something that's like might become cancerous and something that's definitely become cancerous? And how did they do it back then? Looking at those kids, how did they account maybe for over inspection error?
So remember that actually a number of the patients who are included in our study were actually diagnosed with their thyroid cancer, perhaps just in the last decade. And so our study has taken place against this whole background of what you're talking about, this evolution in our understanding of thyroid cancer development and precursors to thyroid cancer, and really this debate about the best clinical approach to handling thyroid nodules. So the complexity of that is certainly one aspect that underlies our analysis, but a fraction of the tumors that were identified in our study were identified through screening. Others were just identified through routine clinical exams, or perhaps even recognition of an enlarged thyroid. But I'm hoping that in future analyses, if we can do a little bit more of the similar genomics work with some of those benign nodules, we'll better understand kind of the genomic progression. But right now, there's not a good predictor for understanding exactly which nodules will go on to develop into a frank malignant tumor.
And what's the delay between exposure and onset of a cancer in thyroid cancers, and then let's say in other cancers as well? Is there a gap between when you're exposed to the radiation and when it becomes a problem?
Yeah, so I think it's really important to differentiate between what's happening in a specific individual versus what we understand from the larger scale epidemiologic approach. And so from epidemiologic studies, we generally think of radiation-related solid tumors of taking at least 5 years or up to decades to develop.
And what's happening biologically in that time period?
Yeah, we haven't known exactly. So what we can tell with this particular analysis is that it appears that the ionizing radiation caused DNA double-strand breaks in key genes that then allowed those thyroid cells to actually transform into tumor cells. But we don't have, we don't look at tissue, for example, that's been exposed to ionizing radiation over time, taking, for example, multiple biopsies in healthy individuals. You could never do something like that. So we don't know exactly what's happening. It could be that for the particular type of tumor, ionizing radiation causes damage, but that damage itself is not sufficient to cause cancer. So that cell is sitting there partially damaged from the ionizing radiation, but waiting for another mutation to occur before it actually becomes a cancer. The other thing that's important to remember, and this is something that we really grappled with in our study, was we don't actually know which tumors arose as a result of radiation. And that's this tension that I was alluding to in the difference between what's happening in an individual patient versus what's happening on the population level. Yeah. So we know that the likelihood that a thyroid tumor arose due to radiation increases with higher exposure, younger age at exposure. But even for an individual who was exposed to a relatively high dose at a relatively young age, we can't necessarily guarantee clinically that tumor actually arose because of the radiation exposure.
And yeah, help me understand just as a reference, what is the background rate of thyroid cancer in a population of this same age group? And then what was the quantity of cancers that we attribute specifically to the Chernobyl event?
So it's really hard to actually nail down those exact numbers. And the way that I like to approach those types of numbers from a radiation perspective is to estimate the relative risk. So what's the factor by which it increases, and that depends on the dose. And so we talk about the excess risk per unit dose, whether that's 100 milligray or 1 gray. And here we're talking about for those individuals who had the highest doses, perhaps they saw more than 4 or 5-fold increased risk. But it's complicated on the population level to actually measure that, again, because there was all of this screening that occurred as well. And so there were some tumors that were perhaps incidentally detected that would never have gone on to become clinically meaningful. And so it's hard to nail down that exact number of saying, well, there would have been X number of tumors, and instead now there were Y.
But just to get a general sense, tell me if I'm wrong, but I've heard something like 1% of the population gets thyroid cancer or something in general, like non-radiation related.
Again, it depends a little bit on age, and
that's still probably 1% would
probably
be
a little high.
The upper limit.
Exactly. And thyroid cancer is more common in women than in men in the general population. And so it's, as an epidemiologist, it won't surprise you to know that I want to get to that who, what, where, when, and why that I was talking about. And so you can make these broad generalizations that sometimes people want to just say, I want to just a number to hang my hat on.
Right, but I'm not looking for an exact number, just to give our audience a sense of the scale of thyroid cancers that are out there today. So 1% is a little high. So we're saying like 0.5% or something?
Yeah.
You could say in that range that would be fine. Again, age is really important in this. And I think what's the difference in Chernobyl was that number is much higher. So depending on the age of the child at exposure and the amount of exposure, the younger the child, the higher the dose, the greater the risk. We've seen from multiple studies. And so it's the kids who were 10 or under who had extensive exposure, who have very, very considerable elevation of their risk of developing thyroid cancer.
And then the population number that we're thinking around for the children that were attributable to the Chernobyl cancer, is that around 5,000 or so? About how many people?
Again, I think it's sort of misleading to focus on a given number, because you have to look at where the accident took place and the spread of the radiation, and that effect wasn't a dichotomous who either were in the zone or not. It just sort of slowly petered out. And what we've learned from the Belarus and Ukraine and Russia, and then there are questions in the Baltic states of much lower doses, but nonetheless, are there thyroid? Is there an excess of thyroid? These are things that we've been looking at very closely, and I don't think anyone would ever want to focus on a particular number. So I'm not going to say 5,000. Because if I say 5,000, it could be 5,001 or 4,009.
No, sorry. I didn't ever mean to focus on a number. I'm looking for general ranges. So I think what I'm trying to get at is like relative numbers. So let's say, 5, okay, it could be 3,000 to 10,000 even. What I want to do is try to understand a comparison of the cohort that got cancer that we think got cancer from Chernobyl versus the total cohort of people who get thyroid cancer, adjusting for the same things, geography and age. Are we saying like around 5,000 compared to around 100,000? Are those numbers about right?
That. So if you think a little bit, if you want to go back to your number of, 0.5% or 1% of individuals get thyroid cancer in a given year, and then, you can essentially say, depending upon the dose level, maybe that risk was increased by, 20% for the individuals with the lowest doses, or more like 3-fold or 4-fold for individuals with the highest doses. But in general, thyroid cancer is still a very rare disease.
Yeah, I mean, we could see from our previous analyses that the relative risk, as Lindsay was pointing out, goes up substantially with higher doses. 3 or 4 gray. Kids under the age of 15 could be as high as 20 times. So we know that there's an excess of cancers. And I would say, Brett, one of the things we have to deal with is we look here, we do these population studies, but we recognize a substantial increase in thyroid cancer was a major problem, a health problem for young adults and individuals in those particular countries. And, it's that tension between what are the statistics in any single case is a problem. And it's a problem for that person and that family. It's 100% or 0% curing of any individual. It's not, oh, we're going to get 70% of an individual. And so when you have in the population level a substantial increase in thyroid cancer above what you would expect, that becomes a major problem. In the Ukraine and Belarus, they established full organizations to search for and take care of the thyroid cancer.
Well, that's actually one of the things I wanted to get to next, and to figure out if that's actually part of the problem that they did that. So I'd like to come back to that screening effect problem. There was a case, have you guys looked at cases of the screening effect taking place specifically in thyroid cancer in other populations? I read one study in South Korea where
we're very aware of that. But again, this is a population that was looked at quite differently because we knew there was this very discrete, profound exposure to radiation.
Right. But so I was going to cite a case in South Korea where they thought there was a nuclear accident. And so they went around in the same methodology and tried to see who had, and they found like a 15 times increase rate of, and so I'm wondering, I mean, that seems kind of similar to the Chernobyl event.
No, they found nodules, not cancer. So big difference. So you have to be really careful what you say. They used a certain apparatus that's very sensitive. And when they went in and looked, they saw no difference in the cancers. What they found was that they were seeing the pre-state or these nodules. And so, you know, again, we want to be scientifically very accurate.
Right.
The comparability is really not there. Okay?
So that's what I was going to ask. So what did they do at the Chernobyl? They took out the thyroid and then put it under a microscope to make sure it was malignant, or how did they do it?
So I think there are a variety of different screening approaches, right? Looking for nodules, and then they made clinical decisions about which nodules needed to be removed. Or actually, as I said, some of the tumors were identified through screening, but others were actually identified just through normal clinical practice. And Brett, one of the things that I think can help is to actually talk a little bit about the thyroid genomic sequencing study that we did, because I think it actually is a totally different way to approach this whole set of questions that can, in some ways, put some of these issues to rest. And the reason is because we had the radiation dose data. So what we did was we did what's called a landscape analysis. So we used a variety of different laboratory techniques to characterize the genome of the thyroid tumors. There were 440 individuals in our study, 359 of whom were exposed after the Chernobyl accident, and then the remainder were unexposed. So they were born after more than 9 months after the Chernobyl accident. And what we were able to do was correlate the radiation dose with the different genomic characteristics. And what we found was that there was an increase in the number of DNA double-strand breaks. We were talking about this earlier, with increasing radiation dose. And there are a number of really important implications of these findings to address these questions that you were just talking about. So we were able to measure DNA double-strand breaks in a variety of different ways. When DNA is broken into strands, what can happen, I often use this image with my fingers so that they can just come back together. And if it's sliced cleanly and comes back together cleanly, then actually there's no damage to the cell. And of course, then there's nothing for us to measure in the lab. What we were able to measure was more when a break had occurred. And in order to put the strands back together, the cell had to take out a few bases of the DNA in order to then do that repair.
Yeah.
Other times what happened is
you lose a knuckle.
Yep, you lose a knuckle. And then other times what happened is there were actually multiple double strand breaks. And so let's imagine there's one break here and one break here, and the cell inadvertently brought together the wrong pieces. And we were able to measure all of those in the lab and then see that the number of DNA double-strand breaks measured in these different ways all were correlated with increasing radiation dose. So the fact that that was our most important finding was really key, because ionizing radiation is not the only thing that can damage cells and cause DNA double-strand.
I was going to ask about that. Does oxygen also do that?
there are a variety of different mutagens that can cause DNA double-strand breaks. And so one of the hopes in our study, or one of the questions we wanted to explore, was whether we could identify a unique biomarker of radiation-induced tumors. And that addresses everything that we were talking about before. How can you know for a specific individual whether radiation caused that cancer? Yeah. And what if, was that just because of an excess of screening or all of these other factors? We didn't find a unique biomarker of radiation-induced tumors because other chemicals or other exposures can cause DNA double-strand breaks. Now, the correlation that we saw between radiation dose and DNA double-strand breaks was incredibly strong. I've described this study as one of the neatest and cleanest set of findings that I've ever worked with in my career, because no matter how we measured the DNA double-strand breaks or other factors, and the results were just all consistent. We often find in our studies, because science is messy and we're trying to push the boundaries of knowledge, that we see some things that line up really nicely, but there's kind of this niggling question of something that looks a little inconsistent and we don't really know what that means. And here it's not that we have all the answers from this one study, but it really was striking when I was doing the analyses to see how well everything really did line up.
And how, yeah, that is pretty amazing. How did how is the various data presented to you? So for both the dose data and for the DNA data, is there another like laboratory somewhere that has sequenced all this DNA and then they ship it to you in a file, or do they ship you guys like the literal actual tissue samples and then you did it with your own DNA sequencer?
Yeah, so we were really lucky for the visionary nature of the scientists who put together the Chernobyl tissue bank, which is currently led by Jerry Thomas.
Yeah, we've interviewed her before as well.
Exactly. And Jerry, in collaboration with clinicians and scientists in Ukraine and Russia, although the tumors for this study came from Ukraine, collected tumor tissues. And importantly, they were fresh frozen tumor tissues. And so by snap freezing the tumors at the time of surgery, we actually were able to have very high quality specimens. And we're really grateful to the patients for their willingness to contribute to the science and to consent to having their tumor specimens used for research. Some of these tumors were collected several decades ago, and there was essentially this understanding that kind of this hope almost for the advances in technology that these tumor specimens would become really important contributions to science. And that's indeed what has happened. And so we received the biospecimens, and then
so they literally physically mailed like frozen packages to you guys.
And we have established collaborations with different labs. We have our own lab and other labs that we work with, pathologists, the people who extracted the DNA and the RNA, and then actually the laboratory scientists who ran all the different platforms that we were talking about.
And then we have 6 or 7 different major types of analyses that were distributed between our lab and a lab up in Boston. And then we had all the files that we were all able to put together with all the dosimetric and epidemiologic data that we had.
And then, so sorry, break it down for me again. So you get the you've got the samples, or you get a sequence, some sort of readout of from the samples, and then how do you determine from that readout what you were saying something about knuckles and double breaks? How do you backtrack out of that?
Well, we get lots of readouts. There's not one readout. We did the little bits of RNA, the long bits of RNA. We did the DNA. We did markers of DNA to look at larger fragments. We looked at the telomeres. We looked at the epigenome. So we had all of this data, and we know what normals look like by and large. And so we did this in each case, comparing either the normal thyroid tissue that was taken out at the time from the other side of the thyroid and or blood. And we make these comparisons, asking the questions, what's somatic? What's new? What does the cancer have? What do we see in this kind of analysis? Okay. And then we could pull those events out and start to organize those. And the real focus of the paper was on what are the characteristics of how radiation and the age of exposure and the age of diagnosis and the actual specific type of mutations in those particular patterns, okay?
And then we're correlating it to the the dose data as well. Did that come on a, like each vial came with, okay, here's what the exposure was, or was it on a, so was it on an individual basis? Like literally they were holding a Geiger counter to it, or was it on like a regional basis based on like soil samples and stuff?
All of the above.
Yeah. It depended on the individual. So for about 20% of the individuals in our study, we actually had some direct measurements. Some of them were based on additionally an interview, others were not. Others were based on direct measurements of individuals who were neighbors or kind of, and that broadened out. And so we had a sense of the strengths and weaknesses of the dose data as well.
And how many samples total did you guys get to look at?
So we included 440 in this analysis.
440 individuals.
440 individuals, yes, of course.
Any more samples?
Were there anything, is there anything in the data that jumped out that we can extrapolate other environmental factors? So for instance, were there two people who maybe like lived close together that had different dose ranges, and then we can interpret like the grass thing that you mentioned, like, okay, this one had a farm that the cows had like a larger area to graze on and that type of stuff?
No, we can't play NCIS to that point of sort of looking at the tumors and saying who lived on the east side of the hill versus the west side. There are some very interesting population genetics questions that is a second wave to help sort of construct what the Ukraine history looks like. We now have these very, very high definition genomes that we can use, knowing things about their family histories. But that's not asking the Chernobyl thyroid cancer question. Okay.
And then one other thing that I heard that I was hoping you guys could comment on was about these children being under iodinized to begin with because of not having proper nutrition. Is that part of the study at all?
We don't have any data on iodine deficiency. And so it's not a factor that we can take into account, but I would certainly say in the last several decades, that's been an active topic of discussion from a radiation accident perspective.
Yeah.
Right. Okay, great. And then in your study, there was also something on hereditary effects and being able to pass on into future generations. Can you talk about that a little bit?
In terms of the thyroid cancer
study,
the second study that you want to talk about, or
the TRIO study?
The TRIO, I'm not sure, because in the thyroid we did look at the germline, what you're born with, and we didn't really see that these children necessarily had a proclivity to develop this due to genetic mutations or a gene profile per se to develop thyroid cancer. If there was, it was very subtle, and we saw it more in the individuals who did not receive radiation. In other words, if radiation is going to come in, it will supersede any genetic effect, is the way we saw that. But are you talking about the second study of the transgenerational?
Yeah, sorry, yeah, that's what I meant to ask about. Yeah,
okay.
Second study.
Because that's a study we conducted in parallel that we set up about 8.5 years ago, where we went to the NRCRM, which is the big center that follows the liquidators, led by academician Bazika. And we wanted to ask the question, was there going to be what we call a transgenerational effect? And to do that, we recruited families. And the way we could do that would be to analyze the entire genomes very carefully in mother, father, and child, and identify if there are what we call de novo mutations, which are kind of building blocks of evolution. So we talked about this before. Every time there is cell replication, there is a small chance of an error. And in every child that's conceived, somewhere between 40 and 70 or 80 of these events occur through the 3.1 billion bases. So when I say 40 to 70 to 80, That's the numerator. The denominator is 3.1 billion bases that could be changed. So we know that there are random errors that take place in the sperm and or the egg, just in healthy, normal individuals. So we wanted to ask the question, if there was radiation to the liquidators and a few of the evacuees who leave, start a family more than 46 weeks afterwards. So in other words, they're no longer at the time of conception in a place of high exposure. Could there be, radiation in either the testes or the ovaries that then damages the sperm and or the ova, that then subsequently increases the risk for more of these de novo mutations? We've known a fair amount about de novo mutations from studies in Iceland and here in the United States and Denmark, where people have done this very high definition sequencing. And so we had some standards and ways to approach this, and we chose to do this at a level that's about 2.5 times more thorough than anyone had ever done. So when we do genome sequences, we talk about running the chemistry of any given base at 20 or 30x in the normal DNA. We did it at 80 to 90x because we wanted to have so much information and be sure we weren't missing or that something very small was lurking, so to speak. And so we did this analysis in 105 families with 130 children, where we had dosimetric evaluation. It was quite accurate. And we actually went back with the dosimeters, dosimetric experts, and re-interviewed those individuals and updated that. And we were able to actually, interestingly, some of the individuals we chose as controls who supposedly had no exposure turned out to have exposures of like 20 milligray, very, very low levels, but nonetheless, they had some kind of exposure that we would need to account for. But similar to our thyroid cancer study, we generated the genomic data, and then we looked at that in the context of a range of radiation exposures that we knew for each of the parents, mother and father. And then we asked the question, were the number of DNMs, de novo mutations in the children, increased in any way in relation to radiation? And the answer was we did not see any evidence for a significant change. We know there's a little difference from individual to individual. We were talking about this before with, genomic repair and stability. But what we did see, which was very reassuring, is that paternal age is very strong. So as the age of the father increases by year, there's an increase at the time of conception of more DNMs. Maternal age is a very small.
And you're saying that has nothing to do with the radiation. That's just in general. Older guys are producing not as good sperm, is what you're saying.
Well, you could say it that way. I have to be very careful how to say this, but we know that with each year,
I'm not offended.
with each year and age added, you have an increase in the number of DNMs. It's small, 1 or 2. Again, these are relatively rare events, but they can, in the setting of, for instance, autism, explain a small but real fraction of autism and other really catastrophic pediatric neurodevelopmental disorders.
And it's funny though, because that's the one thing that people just in like common lore are afraid of from radiation, are these effects being passed on to children. Has there ever, so you guys determined that in the Chernobyl, that was not the case.
We did not see a significant effect. We don't have the precision to say that it never happened.
Got it. Are there any cases of radiation? Because there are only so many big population radiation events. Right. Have we ever seen an increase in these de novo mutations from radiation?
Well, not in the way in which we looked at this. And the reason I'm giving that caveat is that we looked at survivors, individuals that left. We clearly know anecdotally, both after the atomic blasts in Nagasaki and in Hiroshima, that in a very short period of time, there were fetal loss and issues around that, individuals who were captured very quickly. So here, these are individuals that leave and start a family and had moderate, range dose. We didn't, we were not able to identify people with acute radiation sickness who had 4 or 5 gray and then tried to have a family afterwards. That would be a rather unusual circumstance because we know the fatality associated with 4 or 5 gray of total body radiation
like
is quite considerable. And if you survive, there's a lot of morbidity associated with that. So this was really looking at this question of the larger fraction of survivors or people exposed. The levels that we looked at were substantially higher than what has been seen in Fukushima. So we view our results as hopefully very reassuring and helpful to the people of Fukushima, and thinking we know that it's a major issue in returning to those areas, is the risk that childbearing parents have for, will there be an effect on
the perceived risk.
The perceived risk, yes, exactly. I'm saying, but that's been a major issue. And so we, there's been quite a bit of press and discussion in Japan of these papers that hopefully can help, with very important political and social decisions that people have to make.
And then just on that point, and then we got to wrap up, just comparing Chernobyl to Fukushima, Chernobyl, we saw this increase in thyroid cancers amongst the children. Did we see any at Fukushima? Have we attributed any increased risk of cancer to Fukushima?
That's a good question, because the screening issue comes back, in terms of, do you find nodules? Yes, but how much of that is finding of the nodules because you have more sensitive equipment as opposed to the development?
Right. I think at Fukushima, they came in ready with the screening question. And so they did simultaneous tests across different populations that weren't exposed.
And Brett, we know that the incubation period, so to speak, from the time of exposure to developing thyroid cancer, we're only 10 years out from Fukushima. So
that should be enough, right?
That's the start of when we would see that. We see some of our Chernobyl cases occurred earlier than that. But remember, as Lindsay pointed out, many of them are 15, 20, 25 years later. So, I think we have to be careful in jumping to conclusions one way or the other, because we have not passed through that risk period sufficiently to be able to say, there's nothing or there is something. Okay. And there's nothing we can do but rush. We can't rush time. We have to be patient about this.
Right, but we haven't seen any yet. And by Chernobyl, between years 5 and 10, we saw a bunch, right?
We saw some, yes.
We saw some.
Very high dose.
The dose levels are so different that I think part of, again, what's so important about analyses that take dose into account is that I think it really addresses a number of these different factors.
Right. Okay. Well, listen, this is a topic I could talk about all day, so hope to have more time with you guys in the future. But for now, let's wrap it up. Thank you guys again so much for your time.
Well, thank you. It's been a pleasure talking with you, Brent. Thank you.
And initiate at least a new approach to the many difficult problems that must be solved in both private and public conversation if the world is to shake off the inertia imposed by fear and is to make positive progress toward peace.